Hello, Ms. Endicott, and welcome to the rest of my Biology 12 presentation on DNA translation. In this presentation, I'll basically cover the documentation of the product, the rationale for choosing this activity, and the connection to the Biology 12 curriculum. Okay, let's get started. In section 1, I'll basically talk about the documentation of the product. Before DNA translation can occur, a process called DNA transcription has occurred, and I'll briefly summarize that. The DNA has gone through a process called DNA transcription. DNA transcription is a process that involves transcribing genetic information from DNA into mRNA. The transcribed DNA message, or mRNA transcript, is used to produce proteins through a process called DNA translation. DNA is housed within the nucleus of our cells and controls our cellular activity by coding for the production of proteins. The information in DNA is not directly converted into proteins, but must first be copied into mRNA. This ensures that the information contained within the DNA does not become tainted. So basically, my project wasn't on transcription, but before translation can happen, transcription has to happen. And this picture kind of summarizes what that is. An mRNA transcript is made from one strand of DNA containing the information needed to produce a protein in the process called translation. So let's move on to the process of translation. So in the process of translation, the first step is the start codon. The first step of DNA translation process is that mRNA, which was made by the process of DNA transcription into mRNA, is bound to a ribosome, the start codon AUG, at the P site. A transfer RNA molecule with the amino acid methionine and the anticodon UAC has bound to the exposed start codon. The codon UCA is exposed at the A site with the anticodon AGU, and the amino acid serine has bounded to the exposed A site. The two amino acids are close enough to form a peptide bond between them. So as you can see from this picture, the codon on the mRNA would be, it would bond to an anticodon, and that would happen because of a complementary base pairing. And basically, the tRNA molecule with the correct amino acid would only bond with the codon that's on the mRNA, and so it would release its amino acid, which would form a peptide bond with the neighboring amino acid. So let's move on to the next steps. This is more in depth than the peptide bonds. This is sort of the chemistry behind the peptide bonds. As you can see, these two groups would bond to form this type of bond, which is a very strong bond and something that would be hard to break. So like a peptide bond has formed between methionine and serine, and the peptide is bound to the A site. The methionine tRNA leaves, and the P site is now exposed. Now a new tRNA can come and release this amino acid. So as we're talking, I guess the mRNA has now, it's moving through the ribosome, and so as tRNA come release their amino acids, they also leave in search of other amino acids. And bonds, these kind of bonds, peptide bonds, would form between the two amino acids to keep them in chain, which would in turn form a polypeptide chain. So if you look at the next steps, another transfer RNA would basically come in. So the ribosome is now moving along the messenger RNA, one codon, bringing the peptide to the P site. This exposes the A site, and the next transfer RNA carrying alanine is about to bind to the exposed P site. So as you can see, this is sort of a diagram of tRNA. It would come in, release this amino acid, like I mentioned before, and it would bond with the adjacent amino acids, and they would, with a peptide bond, which would form a chain. And just sort of a side note, it isn't always in this order: alanine and methionine, serine, that kind of thing. It's just the specific one we're talking about is in that order, and you would know because of what the message in the codons, on the mRNA. So let's move on to the next step. This is sort of the elongation steps. As you can see, the mRNA has moved down the ribosome has moved along one codon down the mRNA in the 5' to 3' direction. This is achieved by the translocase enzyme, and the process of ribosome translocation continues. The old tRNA is released to bind to another amino acid and go in search of a new codon. the binding of the new amino acid is mediated by an enzyme called amino-allyl tRNA synthetase. So as you can see, this process, like I said, will continue on, and the chain would just get longer and longer with more amino acids bonding, and it would happen at a very fast rate. So this picture kind of summarizes it. You can see the tRNA are bringing their amino acids, and they have their anticodon, which would bond to the mRNA. If it's the correct one, it would release its amino acid, and it would bond with a peptide bond to form a longer chain until, of course, a stop codon is reached. So this is sort of the termination step. The process that I mentioned before would continue on for a long time, depending on how long the protein needs to be or how long the amino acid chain needs to be. But basically, if we fast forward, a termination step would happen. The process would continue along the mRNA until a stop codon is reached. While there's no tRNA for stop codon, there's an enzyme called release factor, which will release the polypeptide chain, resulting in a new protein. So to summarize the termination steps, finally the complex is disrupted, the ribosome separates, and the mRNA is released to be used again or degraded. Translation occurs at multiple sites along the mRNA so that many ribosomes can be seen by electron microscope bound to a single mRNA strand with many polypeptide chains forming in each. So basically this picture kind of summarizes it. As you can see, the codons will act as stop signs, kind of signifying the end of the protein chain. And like I mentioned before, this would happen at multiple states along the mRNA, so just because one stopped doesn't mean all of them would. So sort of a general overview of protein synthesis. As you can see, DNA transcription produces mRNA, and mRNA through translation produces proteins. So this will kind of sum it up. My project, like I said, wasn't specifically on transcription, but I guess to summarize transcription, the purpose of it is to make mRNA. The mRNA will hold codons that tRNA will translate into amino acids, and to translate, to kind of summarize translation, the purpose of it is to make polypeptides, or in other words, proteins. And proteins are absolutely necessary for your body. Proteins can be used as enzymes to catalyze reactions, or as transporters to help nutritions move in each and every cell in your body. So in section 2, I'll kind of talk about the connection to Biology 12. Biology 12 is a dynamic course filled with many interesting details. Protein synthesis, and specifically the process of DNA transcription, is one of the more difficult aspects of Term 1 of Biology 12. And after completing this Term 1 choice assignment, I now have a deeper understanding and a more complex understanding of the process of DNA translation. So that was kind of my connection to the Biology 12 curriculum. In section 3, I'll kind of talk about the rationale for this activity. The reason why I picked this activity for my Biology 12 choice assignment was because I believe that the human body is a complex machine with many parts and many functions, but if you break it down, it all comes down to genetics, and the process of how it's replicated is important to the rest of our body and sets the foundation for a study of more complicated aspects of biology. Personally, when you look at aspects of our body that are so microscopic, you can't help but think of our Creator, and this is, I guess, a verse I put in here. This is Psalm 139 verse 14, which basically says, "I praise you, for I am fearfully and wonderfully made. Wonderful are your works. My soul knows it very well." So just basically just says that, every time, you look at things like this, at least personally, I can't help but, stand in awe at, how complex the body is. So that's kind of why I put that in there. And yeah, I think that's basically my presentation. So thank you for watching, and I hope you have a nice day.